Hello students, welcome to EPG Pathshala. I am Dr. Hidayatullah from Department of Biophysics, All India Institute of Medical Sciences. Today we are going to discuss about a model, structure of protein-ligand complexes under paper biomolecules and their interactions. This model will discuss about knowledge gained from structure of protein-ligand complexes like mode of ligand interactions, specificity of ligand, and the protein recognition for specific ligands or substrates. For example, enzyme-drug complex, transporter carbohydrate complex, ATP binding protein complex, receptor ligand receptor drug complex, enzyme antibiotic complex, and their resistant mechanism. All the fundamental cellular process is mediated by an array of biological signals. These biological signals are mediated by ligands such as nucleotides like adenine triphosphate, ATP, guanine triphosphate or GTP, or amino acids like glutamine, leucine, which are important in glutamine transporter, leucine transporter, ions like sodium, potassium. We have channels like sodium channels, potassium channels, chlorine channels, then carbohydrates like maltose transporter, glucose transporter, sucrose transporter, etc. There are also small molecules which are important for mediating the ligand interactions. So how do we define ligand? A ligand can be defined as a molecule which can trigger a conformational change in a protein or any other macromolecule which lead to an array of biological signals. Importance of protein-ligand complexes. Complex arrays of intramolecular interactions depends on the binding site of the macromolecules. It is also depends on the interacting ligands. So the knowledge we gained from the structure of protein-ligand complexes are mode of binding of ligand, solvent around the binding site or active site, the mechanism of enzyme activity or protein function, flexibility of protein binding site, optimization of drugs to develop new compounds. Enzyme drug complex. So here we see an example for phospholipase A2 complex with non-steroidal anti-inflammatory agent aspirin. So phospholipase A2, it is an erythrolytic enzyme, binds to the glycerophospholipids and hydrolyzes the lipid to liberate pro-inflammatory substrates like arachidonic acids, which leads to the inflammations. These arachidonic acids are further processed by the inflammatory pathway enzymes and they produce a substrates called prostaglandins, which are called as eicosanoids. These eicosanoids are the ones which cause a physiological effect on the disease like arthritic rheumatoids and asthma. So there are people who are researchers working on these enzymes to design drug against this enzymatic pathway. So acetyl salicylic acid, aspirin, a well-known drug often used to treat pain and inflammation. This acetyl salicylic aspirin, it is extracted from the willow tree before we know about the pharmacological effect of this drug. This aspirin has been previously used against these inflammatory pathway enzymes. Enzyme drug complex with PLA2 with substrate. PLA2 specifically cleaves phospholipids at SN2 position. So lipid binds at the hydrophobic channel. So in this figure you see the PLA2 along with the lipid. The lipid is shown in sticks in green color. The active site amino acids histidine, aspartic acid and tyrosine are shown. And you also see that the lipids interacts with the aspartic acid and histidine. That's how the catalytic reaction happens. On the right side you see a surface representation of the lipid and the PLA2. So the lipid binds in the hydrophobic cavity of the PLA2. So any inhibitor or ligand design should basically block the active site or the hydrophobic channel to prevent the catalytic reaction or the binding of the substrate. Here we see enzyme drug complex with the aspirin. The anti-inflammatory agent aspirin binds to the active site of the PLA2 by forming water-mediated interaction with the catalytic amino acids histidine 48, aspartic acid 49 and calcium ion. Here we see the cartoon representation where the aspirin is shown in sticks in cyan color, the calcium ion is shown in blue color, and the active site amino acids are shown like histidine 48, aspartic acid 49. So you see that aspirin is interacting with the catalytic amino acid through a water mediated interactions, and it forms a direct interaction with the catalytic calcium ion. On the right side you see a surface representation where aspirin is completely buried inside the active site channel. So it is buried inside, that's how it inhibits, but does not completely inhibits because it's not covering the whole substrate binding channel. So aspirin takes over the place of OH group of lipids as you seen in the substrate complex, but it does not block the complete hydrophobic channel. So there is a scope for improving the inhibitors in this. From this in this complex we can understand that there's a scope to improve the inhibitor complex. So as we see from the model, we understood that how the PLA2 drug complex was being made with non-steroidal anti-inflammatory drug aspirin. So to design more specific inhibitor for phospholipase A2, we need to block the hydrophobic substrate binding channel. So to block the hydrophobic substrate binding channel, you need to design inhibitor which should be long hydrophobic, and it should also interact with the catalytic amino acids. So from the example of aspirin, we know that the inhibitor should have a OH group that might be an ideal candidate to form interaction with either water mediated or direct interaction with the active site amino acids like histidine 48, aspartic acid 49 of PLA2. In short, the inhibitor should have a hydrophobic group so that it can block the hydrophobic channel as well as the active carboxyl group or an OH group so that it can directly interact with the active site amino acids like histidine 48 and 49. So transporter carbohydrate complex. So glucose is an essential fluid for all biological organisms. In human there are about 14 types of essential glucose transporter called as GLUTs. So all these GLUTs, they belong to SLC family, that means solute carrier type transporters. There are almost 14 types. In these 14 types, GLUT1 to GLUT4 are the most essentially studied transporters so far. GLUT1 transporter is responsible for transporting the glucose across the blood barrier, blood tissue, whereas GLUT2 transporter is responsible for transporting glucose across kidney, intestine and other organisms in liver such as. GLUT3 transporter is mainly responsible for transporting glucose across the brain for giving glucose to the neurons. So each type of GLUT transporter has a specific function at a specific site in body. So mutation in this glucose transporter like GLUTs leads to some diseases like GLUT1 deficiency syndrome when there is a mutation in GLUT1 transporter, type 2 diabetes mellitus which is in mutation in the GLUT2 transporter, and when there is a mutation in the GLUT3 transporter then you have Alzheimer's disease, etc. So in this module we will be discussing about the structure of complex of GLUT3 transporter in complex with alpha D glucose, GLUT3 transporter with beta D glucose, GLUT3 transporter with maltose which is also an exophageal inhibitor, and we'll see how the structure explains the substrate specificity for this sugar and the selectivity by the GLUTs. Transporter carbohydrate complex, GLUT3 glucose transporter. GLUT3 glucose transporter facilitate diffusion of glucose, and it is responsible for glucose supply to the brain and other organs. The structure shown here is a GLUT3 transporter consists of a canonical major facilitator superfamily fold with N and C domain enclosing a cavity that opens towards the extracellular or outward open conformation. What I mean by extracellular or outward open conformation? The figure on the right side, what you see is a GLUT3 transporter. So you see N-terminal domain in violet color. It consists of 6 alpha helical domain. On the right side you see yellow color 6 alpha helical domain, which is a C domain. So at the center you see a small ball and stick, which is a glucose. So the glucose is bind at the center of the alpha helical domain, N-terminal domain and the C-terminal domain. So when the glucose is at the cytoplasm, it gets transported into the periplasm through this transporter. So now you see that GLUT3 is open towards a periplasm. So when it is open towards the periplasm, we call that as an outward open conformation. If it is open towards the cytoplasm, we will call this inward open conformation. Here we see that GLUT3 transporter complex showing that how GLUTs bind to A or beta anomers of D-glucose. That means alpha D-glucose or beta D-glucose. In the left hand side you see two figures, right? Here in that one we see that alpha beta glucose bound which is shown in violet color. So you see that the tryptophan recognizes the OH group on the left side. On the right side you see same beta D glucose where tryptophan also recognizes the sugar. The difference between the alpha D glucose and beta D glucose is shown on the right side. So where you have stereo projection, right? In this one you see in alpha D glucose the OH group is pointing downwards, whereas in beta D glucose the OH group is pointing upwards. So now come back to this figure on the left side. So here in the violet color you see that the OH group is pointing downwards, which is alpha D glucose, and it's been recognized by the tryptophan. On the right side, which is in the glucose in the gray color, you see that OH group is pointing downwards. So that's how this GLUT3 transporter recognizes this both anomers of D-glucose. That means it can recognize both alpha D-glucose also and beta D-glucose also. That means it can transport both type of glucose into the cell. Based on the knowledge of structure complex, the researchers tried to study different sugars with both A or beta anomers. So can the GLUT3 can recognize other sugars also? So they have done extensive study with different types of sugars which are shown on the left side of the figure. So you see D-glucose, L-glucose, D-fructose, D-galactose, L-galactose, like that. So the sugar which are marked in with a box are having a very good transport by the GLUT3 transporter when they did the periplasmic based transport assay. Whereas the glucose which are shown in bold like L-arabinose, L-lyose are showing a slightly inhibitor effect on these GLUT3 transporters. So this observation is very complicated. So the important thing is that how is it able to recognize OH group? In one complex they are able to recognize the OH group. In certain complexes when they did the biochemical transport assay, you see the inhibitory effect. So the important observation from the complex is that in general the aromatic residues in the active site are considered to be involved in stacking interaction, whereas the structure complex of GLUT3 with the glucose showed that the tryptophan, which is an aromatic amino acid, tryptophan 386, is involved in recognition of alpha beta form of sugars. So as we see in the previous slide that they tried different types of sugars to see how it can transport the sugar across the membrane. So they were puzzled by the different sugars showing different specificity and different transport mechanism. So the researchers went on to make a complex of disaccharide, which is a maltose. Maltose is also considered as an exofacial competitor for D-glucose. So it is a longer sugar like disaccharide. So it's a dimer of glucose. It also binds to these GLUTs, but the transport is not as efficient as compared to D-glucose. So they made the complex of GLUT3 transporter with the maltose sugar, and the structure shown here in the cartoon representation is shown the glucose transporter, GLUT3 transporter with the maltose, and it is also an outward open conformation. It is open towards the periplasm. So the maltose is shown in sphere at the center of this domain. Okay, so on the right side you see two figures, where one you see the interaction of maltose, which is shown in cyan. So here also the tryptophan is recognizing the sugar, which is the alpha D glucose, and it is also, you see that the disaccharide is very longer. On the right side you see the superimpose of D glucose complex and the maltose complex. So here you see critically two difference. The one in the green color belongs to that of complex of GLUT3 complex with maltose sugar, and which is shown on yellow is the complex of GLUT3 with glucose. So here you see that particularly the amino acid which is shown in the arrow is asparagine. In case of D-glucose, the asparagine is moving towards the glucose and it is interacting. This belongs to a transmembrane helix. This asparagine belongs to a transmembrane helix. When glucose is there, then the transmembrane helix can move. Whereas when the maltose is there, you see that asparagine is moving away. It is shifted away. So the transmembrane helix cannot move. It is basically the another disaccharide is blocking the movement of the transmembrane helix. So as a result, there is a slow transport, and it's also, we can also call it as an inhibitor. So we saw example for GLUT3 transporter with alpha beta glucose, alpha beta D glucose, and beta D glucose, and maltose complex with the GLUT3 transporter. So the knowledge we obtained from the GLUT complexes is that the GLUT3 complex reveals that the GLUT can recognize both anomers. That means beta D glucose also it can recognize, alpha D glucose also it can recognize. Hence anomerization may not be required for the D glucose transporter by GLUTs. Either it can GLUT1 to GLUT4 or GLUT12. So anomerization is not required. It can recognize beta D glucose also, alpha D glucose also. Whereas when you have a longer disaccharides like maltose, xylose or other disaccharides, these disaccharides basically hinders the transition movement, the facilitated diffusion of the glucose transporters. As a result, the transport becomes very slow. The transport specificity becomes very slow. So the GLUT transporter has become an, for this knowledge which we gain from this structure can be utilized to design drug against this GLUT transporter, because the GLUT1 transporter is also an attractive target to block glucose uptake in malignant cells. So if we design any inhibitor which can slow down the uptake or it inhibit the function of the GLUT, then we can kill the malignant cells. Hence, using the structural information, we can screen for inhibitors which are longer, which can block the transport. Either it can be a sugar inhibitors or it can be a molecules which are hydrophobic, which can recognize the particular tryptophan 386 which we see in the structural complex of GLUT3 with glucose transporter and maltose transporter. So it can block the C-terminal transition to transport glucose diffusion across the membrane bilayer. So from these complexes, it reveals that the alternate access transport mechanism, which is very important for transporting the glucose across the biological membranes. So to understand, students, to understand the mechanism, I'd suggest you to watch the animation video in the supplementary section in these articles which was published in Nature. So go through that supplementary section, see the animation video, understand how the transition happens when the sugar binds to the glucose transmitter, how it transports from the inward conformation to the outward open conformation. Inward conformation means it opens towards the cytoplasm. Outward conformation means it is open towards the periplasm. So when the sugar is there in the cytoplasm, in the inward open conformation, the sugar comes and binds to the cavity. This binding of this sugar induces a conformational change in the transporter. As a result, the sugar get transported towards the periplasm. This is called as the diffusion mechanism in case of GLUT transporters. I will suggest you to go and watch the video in the supplementary section in the article and understand the glucose transporter mechanism for the GLUT transporters. Here we see the maltose transporter ABC binding domain with ATP. So ABC family proteins bind ATP. Upon ATP hydrolysis, use the energy to drive the transport of various molecules across the plasma membrane as well as across the intracellular membrane. So this ATP binding proteins or ABC family proteins, they consist of multi-subunit. One is the ATP binding cassette associated with two nucleotide binding domains as a cytoplasmic portion and two transmembrane domains to translocate substrate or essential nutrients across the membrane. On the right side you see a cartoon representation of a nucleotide binding domain from maltose transporter. So here on the left side you see a one monomer which is in green color. On the right side you see one monomer with cyan color. So in the center you see sphere representation. There are two ATP molecules. So here the ATP molecules are helping in dimerization of these nucleotide binding domains. When these two nucleotide binding domain dimerize, they open the transmembrane helices. So when they open the transmembrane helices, the substrates like here in case of maltose transporter, the maltose gets inside the transmembrane helices. When the hydrolysis happen at this nucleotide binding domain, they again move away further away. With the movement of this nucleotide binding domain makes the transmembrane domain to close. As a result, the maltose gets transported across the transmembrane. As we see there, you have a dimer of ATP in case of maltose transporter. So nucleotide binding domain, as I said earlier, dimerize when they bind ATP. It is also referred as closed dimer. So the complete ATP binding site is formed when nucleotide binding domain dimerize in case of ABC family proteins. So in general, two ATP molecules get sandwiched between the conserved Walker A motif of one monomer. Walker A motif has a sequence patterns like G, X, 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 X followed by G or K followed by T or K. And X can be any amino acids. So Walker A motif from one monomer and ABC signature motif, which is nothing but a sequence LSGG motif. So of other monomer, they help them to dimerize. So in this figure on the left side you see a monomer of a nucleotide binding domain where you see ABC domain which is in green color, regulatory domain which is on the cyan color. So this ABC domain has two domains. One is the alpha helical domain and other is called Rec-A domain. So Rec-A domain is the domain where the ATP comes and binds. So on the right side you see a zoomed image of the ATP bound to both bind to the dimer. So here you see the Walker A motif which is shown by the arrow. So it has the GXXXX G/K motif. So you see the beta phosphate of the ATP bound at the dipole helix. On the right side you see the LSGGQ signature motif coming from other monomer. This LSGGQ signature motif creates a motif which is followed by a helix loop helix motif. That is, the 310 loop is formed because of this particular LSGGQ. So this provides interactions to stabilize the ATP. Apart from that, you see a tryptophan on the left side which is shown in green. So this tryptophan provides a stacking interaction with the adenine group of the ATP. So this places the adenine group very stable until the hydrolysis happens. Once the hydrolysis happens, the gamma phosphate get released, and as a result the dimer opens up, which results in opening of the transmembrane to transport the maltose across the membrane. Enzyme with ATP binding domain. Muri ligase with ADP/substrate UDP N-acetyl-myramyl L-ala-D-glu. Most of the Moore ligases are ATP dependent. ATP dependent Moore E is an essential enzyme for the synthesis of cell wall peptidoglycan in bacteria. Moore E ligase folds into three domains: N-terminal domain, which is shown in red on the right side, ATPase domain or ATP binding domain, which is shown in yellow on the right side, C domain or catalytic domain, which is shown in violet on the right side. So it binds to the UDP N-acetyl-myramyl L-ala-D-glu, which is shown as a stick on the right side, and it adds the mesodiamino-pimelic acid in case of gram-negative bacteria or lysine in case of gram-positive bacteria. So when ATP binds to this ATPase domain, after hydrolysis, the catalytic domain comes closer to this of substrate, then that mesodiamino-pimelic acid added as a tripeptide is added as a third peptide to this substrate, or it is a lysine in case of gram-positive bacteria. Moore ligase can differentiate UDP and ATP. So ATP binds to the ATP binding domain, whereas the UDP substrate binds only at the N-terminal domain. So as we observed in N-terminal binding domain of ABC family protein in previous section, Moore ligase also has a typical ATP binding motif where ATP binds, but it is not involved in dimerization, whereas it is involved in domain movement. Upon hydrolysis, the energy released from the ATP is utilized for domain movement in case of Moore ligase. So in the ATP binding protein examples, we saw two examples. One is the maltose transporter nucleotide binding domain where ATP binds and it dimerizes. So a second example is Moore ligase where it uses the ATP to bring the catalytic domain closer to the top ATP domain so that the third amino acid like DAP or lysine can be added to the pentapeptide peptidoglycan. So to conclude, ATP or GTP binding requires Wakare motif, that is nothing but a P loop or phosphate loop where the beta phosphate goes and binds to the beta phosphate of ATP binds with a sequence pattern binds to the nucleotide binding domain, and this phosphate loop has a sequence pattern called GXXXXG. It can be glycine or a lysine followed by T, threonine or lysine. So it can be any amino acid residues which can follow this pattern. This motif is formed at the start of the dipole helix, which is very important for the beta phosphate to bind to the beta phosphate of the nucleotide bind strongly to the nucleotide binding domain. Once this ATP binds strongly to the nucleotide binding domain, in case of maltose transporter nucleotide binding domain, they induce a dimerization in the nucleotide binding domain. Whereas in case of Moore ligase, this binding of ATP brings the catalytic domain closer to that of the ATPase domain so that the third amino acids can be added to the pentapeptide. For the nucleotide binding, another important feature is that stacking. For nucleotide stacking, aromatic amino acids like tryptophan, phenylalanine, tyrosine is always observed in the ATP binding region. So depending upon the protein, after ATP hydrolysis, the hydrolysis energy will be released. Energy will be released by ATP hydrolysis will be utilized for conformational change or catalytic reaction. In case of maltose transporter, it is required for a conformational change, which will open up the transmembrane region in case of maltose transporter. Whereas in case of Moore ligase, that ATP hydrolysis of the ATP is used to bring the catalytic domain closer to that of ATPase domain so that there's additional peptide like pimelic acid or lysine can be added to the tripeptide UDP acetyl-myramyl tripeptide. Signaling in neurons is controlled by serotonin receptors. The major function of the serotonin receptor is to translocate the serotonin from the synaptic cleft back into the presynaptic terminal. Due to this SLC6, that is means solute carrier 6 neurotransmitter A4 polymorphism, there is a change in the rate of serotonin uptake which leads to the aggressive behavior of patients having Alzheimer's disease and post-traumatic stress disorder. Hence, antidepressants are prescribed to these patients. These drugs are also called as selective serotonin reuptake inhibitors (SSRIs) as they bind directly to the serotonin transporters. But the problem here is that with these drugs, as these drugs also binds to other homologous receptors like norepinephrine and dopamine transporters. There is no crystal structure so far for the serotonin receptors, but there is a homologue of serotonin receptor which is LU-T transporter, which is called as a serotonin homologue, which is shown on the right side figure. So you have the serotonin LU-T transporter with a leucine substrate bound at the cavity of the LU transporter. LU-T is an homologue for human sodium importer family. LU-T transporter from Aquifax Aleocus shares 20 to 25% sequence identity with human serotonin transporter, norepinephrine, and dopamine transporter members of sodium importer family. The crystal structure of LU-T, which is shown on the right side in violet color, its transport mechanism is considered as a good model system for the study of mammalian NSS family proteins. So on the right side you see the cartoon representation of LU-T transporter with the leucine bound at the center of the cavity, which is shown in ball and stick. Here we see crystal complex made by the group with different amino acid substrate which acts as inhibitor. So we have four structures here: leucine complex with the LU-T transporter, tryptophan complex with the leucine transporter, alanine complex with the leucine transporter, and PPF, which is an inhibitor of leucine transporter. On the right side you see the cartoon representation of LU transporter with the leucine bound at the substrate. This is the same position where all these amino acids binds. Here very important region is that you have a carboxyl group in each amino acids. This carboxyl group is neutralized by the sodium, which is shown in blue color in all these substrate complexes. The drug sertraline binds to the extracellular visible region in LU-T. This region is close to the halogen binding site in LU-T transporter. The drug binds to the extracellular side, inhibits the movement of transmembrane helices, which is shown on the arrow. You see the serotonin bind at the extracellular visible region. The drug forms an halogen binding packet and blocks the LU transport. The drug has a chlorine ion as an halogen, which binds to this halogen binding site and prevents the movement of these helices. The fluoxetine binds to the extracellular visible region in LU-T, which is shown in yellow on the left side, which is bound to the extracellular visible region. On the right side you see the zoomed image of that binding of fluoxetine to the LU-T. This region is also close to the halogen binding site, which is observed in serotonin also. The drug binds to the extracellular site and inhibits the movement of transmembrane helices. The drug forms halogen binding pockets similar to that of sertraline, blocks the LU-T transport. This fluoxetine has a fluoride as a halogen. Both sertraline and fluoxetine binds to the same extracellular visible region in LU-T. Binding of drug to the extracellular site inhibit the movement of transmembrane helices. Drug specificity is majorly depends on the halogen group forming a halogen binding pocket. Biochemically and physiologically, it was observed this antidepressant binds to other norepinephrine and dopamine receptor. Enzyme antibiotic drug complex DNA gyrase quinola. DNA gyrase is a type 2 topoisomerase. Functionally, it forms heterotetramer A2B2 gyrase A gyrase B, which is shown on the right side. The primary function on DNA gyrase is to introduce negative supercoil during DNA replication process. Basically, it cleaves the double-stranded DNA, which is shown on the figure, at one end and pass through other strand, thereby introducing negative supercoil. Quinolone, known antibiotics used as an inhibitor against DNA gyrase. Ciprofloxacin, which is in gray, stacks with DNA, which is in orange color, interacts with the serine 83, which is shown in yellow on the left side, thereby inhibit the supercoil process during DNA replication. The quinolone drug inhibition depends on the magnesium ion concentration, which is required for the effective inhibition of topoisomerase. Fluoroquinolones antibiotics was used for the treatment of enteric fever, which basically targets DNA gyrase, as we observe from the complex how it inhibits. On the left side you see wild type where the fluoroquinolone antibiotics interacts with serine 83. On the right side, due to the mutation of aspartic acid 87, it gets uncoiled, which is shown in cyan color, so it loses the interaction with the antibiotics. So the bacterial passengers has gained resistance by introducing simple mutation at the quinolone binding site like serine 83 to phenylalanine to tyrosine or aspartic acid 87 to glycine as shown in the figure, which drastically reduce the affinity of these drugs. As a result, the organism gains resistance. Lock and key hypothesis. In 1894, Hermann Emil Louis Fischer put an hypothesis that a substrate and an enzyme have specific geometry, shape that fits exactly into each other, having a natural geometry called as lock and key hypothesis. It was assumed to be applicable to all protein substrates. So in the figure you see that there is a shape which is designed for the substrate, which is shown as S. So the substrate basically goes and binds exactly to the shape. That is the lock and key hypothesis proposed by Hermann. The lock and key hypothesis basically failed in some cases. So there is another hypothesis which was proposed, which is called induced fit hypothesis. In actual biological scenario, enzymes change their shape slightly to match the substrate. Hence, another scientist named Daniel Koshland slightly modified the lock and key hypothesis to induced fit hypothesis. The hypothesis says that the enzymes are flexible, the active site is constantly being reshaped by the interaction with the substrate. So students, let us summarize what we have learned in this model. In this model, we have seen some examples with lock and key hypothesis and induced type of ligand protein complexes. For example, lock and key hypothesis, we have payload 2 where the lipid comes and binds and it gets hydrolyzed into certain eicosanoids. In case of induced fit hypothesis, we have seen that in loop transporter, we have leucine binds to the transporter which induces a conformational change in the leucine transporter. We have also seen in the glucose transporter where the glucose comes and binds, it induces a conformational change so that it gets transported across the membrane. We have also seen example for the LUT along with the drug complexes. So these are induced fit type of ligand protein complexes. We have seen that this hypothesis, this induced fit, the lock and key hypothesis also fails in case of some cases like receptor drug complexes, where you see we observe that the drug can bind and tightly and it will inhibit the function of the protein, but it is binding as a different extracellular region, so their lock and key hypothesis fails. With examples used for drug receptor complex or enzyme complex, you have learned that how drug can be designed to bind to specific cavity or a binding site. For example, in In payload 2, we have seen that the drug aspirin binds only at a certain portions, but it does not cover the hydrophobic channel. For example, in case of complex of payload 2, there is a scope which will improve the drug design. Like you can design more hydrophobic inhibitor which can block the hydrophobic channel, and also it specifically bind to the active site amino acids to become more specific to a particular target. But the lesson learned here is that biochemically we can find any drug interacting with the target molecules. You can screen thousands and thousands of compounds to bind to the molecule. But actual atomic detail will help us to improve the specificity and also avoid the promiscuous drug which leads to the side effect in case of human.